Thanks everyone for joining. Really great to see a lot of familiar names, and also to see some folks who looks like maybe are newer to our community. So I want to just extend a warm welcome to everyone. For those maybe who have recently joined the chordoma community, my name is Josh Sommer. I'm co-founder and executive director of the Chordoma Foundation. I'm a 16-year chordoma survivor, and this is a kind of a special moment because I'm just recently back to work after surgery last month to resect a local recurrence of my clival chordoma, and thankfully I am doing great. Surgery was quite successful. The whole tumor was removed, but certainly it definitely brings a heightened sense of urgency to the work we're doing at the foundation, to the work that all of you are doing, and certainly only adds to my appreciation to for all that you all are doing to advance chordoma research. So I think in that spirit, I'm really glad that you all have joined us for this introduction to CF Labs. Really excited to fill you in today on some of the initial progress we've made in standing up the lab, the capabilities that the lab offers, as well as our vision for how the lab can potentially serve as a resource to you going forward. And as I mentioned, just as a patient, it is just extraordinarily encouraging to see the speed and efficiency gains that the lab is already making possible in a lot of key areas. And my hope is that in time, the lab is going to help you all, many of you, answer questions or pursue ideas or take advantage of opportunities to drive forward the discovery of new and better therapies. So with that being said, I'd now like to introduce my colleague, Dr. Dan Fried, who is the founding director of CF Labs, as well as our head of target discovery and translational research, who's going to be leading today's webinar. For those who don't know Dan, we were fortunate to recruit him to the foundation at the beginning of 2021, basically to lead our entire preclinical research portfolio. So everything from model development to target discovery to translational research. And Dan has a very impressive background that I think is really ideally suited for this role, having spent more than 15 years in basic research, as well as translational and drug discovery research spanning academia and industry. So before I turn it over to Dan, I want to share a bit about his background, which I think is quite relevant to those who might be considering collaborating with Dan in the context of CF Labs. Just briefly, Dan earned his PhD in chemistry from UVA. From there, he moved on to Penn for a postdoc, where he studied cell signaling by receptor tyrosine kinases using a variety of approaches, including biochemistry, biophysics, and structural biology, and collaborated with medical oncologists to understand the effects of specific cancer mutations on kinase inhibitor sensitivity. From there, he completed his training at Yale's Department of Pharmacology and Cancer Biology Institute. And after publishing several high-profile papers, he took a position at a biotech company here in North Carolina called G1 Therapeutics as the lead translational scientist on their oral CDK4/6 inhibitor program. At G1, Dan evaluated various combination therapies and preclinical models across different indications in order to develop rationale for new clinical trials. And his job was really highly collaborative. He partnered with scientists at academic institutions and pharma companies to test a variety of specific therapeutic hypotheses. And ultimately, Dan was part of a team that secured 3 INDs and FDA approval for their drug trilaciclib. After G1, Dan was on the scientific founding team at a startup called Xylus, where he helped develop the proprietary organoid technology, leading to a $70 million Series A financing round. And now at Chordoma Foundation, Dan draws on all of these experiences to develop research enabling infrastructure, everything from our biobank to model repositories to drug screening program, to catalyze discovery and validation of new targets, to collaborate with various research partners, and ultimately to generate a strong preclinical data package to justify new clinical trials for chordoma. So I'm really, truly grateful to have Dan on our team here at the foundation. And as a patient, I feel just extremely fortunate to have him working day in and day out to help advance discovery of better treatments for this disease. Dan, without further ado, I'll turn it over to you.
Okay, let me try to share my screen here. Josh, is that showing up for you?
Yes.
Perfect. Okay, thank you, Josh, for that kind introduction, and welcome everyone, and thank you for joining us today. I'm excited to share more with you about the launch of our new lab, and really also to just kind of start a conversation about how we can best position ourselves to be a helpful resource to both the chordoma research community as well as the patients that we serve. So before we get started, a few housekeeping notes. This webinar is being recorded and it will be available online if you want to run through it again, or if you have any additional questions after the fact. We do welcome questions throughout the webinar using the Q&A button, so feel free to send those in at any point, and then we'll get to those at the end. And then subsequently, if you have any additional follow-up questions or feedback at the end, please do reach out to us at support@chordoma.org. So as some of you have probably heard through our communications and on social media, the launch of Chordoma Foundation Labs is probably the most consequential step that we've ever taken to advance our mission. And we're really excited to share the motivation for taking this step and where we're at in setting up the lab, as well as to give a view into the impact that it's already having through some of the projects that are currently underway and our vision for the impact that the lab can have moving forward. So we have an hour allotted for this conversation, but I'm available to stay on for a bit after the hour if additional questions come in. So please don't hesitate to submit those using the Q&A button. So to provide some context, it's worth starting with what our guiding mission is, which is to improve the lives of those affected by chordoma and to lead the search for a cure. And we recognize that in order to carry out that mission, a number of things are important. So we need to invest in research to lead to better treatments, in healthcare improvement to foster better care, and in patient services to deliver a better experience. And together, each of these initiatives are mutually reinforcing and ultimately allow us to move towards a future where patients can overcome this disease and maintain their quality of life. And what we're going to be focusing on today is ways in which we can catalyze chordoma research. So as many of you know, surgery and radiation are the cornerstones of chordoma treatment and have really advanced significantly over the past few decades and become quite sophisticated, and in many cases, incredibly effective. A number of patients can achieve several years of disease-free survival, and in some cases are even cured. But unfortunately, the majority of chordoma patients still experience a recurrence, and at the same time, surgery and radiation are frequently associated with significant quality of life impacts arising from the fact that these tumors grow in sort of high-value real estate locations anatomically. For patients that do experience disease recurrence, the treatment options are currently incredibly limited. At some point, surgery and radiation are no longer safe or feasible, and consequently, the median survival for chordoma remains around 8 years. So what's needed? Frankly, we need better treatments that can eliminate or durably control advanced disease, that can prevent or minimize the likelihood of disease recurrence, and improve the quality of life. So towards that end, there are really extraordinary advances that are happening throughout the field of oncology, as many of you know, that are leading to opportunities to improve treatment and outcomes through better detection, more advanced local therapy approaches, including surgery and radiation, cutting-edge systemic therapies, as well as advances in personalized medicine. And among these various options, the most urgent need is in systemic therapies that can halt disease progression and allow patients to maintain their quality of life when local therapies are no longer safe or effective. So multiple potential paths can lead to better treatments. One path, which represents the fastest path to changing the treatment paradigm involves repurposing existing drugs. So these can either be drugs that have already received marketing approval or investigational drugs that were initially developed in different disease indications. And so we have a high interest in understanding what chordoma's vulnerabilities are and whether it shares vulnerabilities with other cancer types so that we can identify drug repurposing opportunities and advance those towards the clinic. Another potential path involves the development of new drugs altogether. So, for example, target discovery studies may identify a novel target in chordoma or one that is unusually enriched in chordoma compared to other cancer types that may lead to new drug development campaigns. But to understand which of these paths can ultimately lead to better treatments, ultimately we need to be guided by the biology of the disease. So there's quite a bit of basic or target discovery research that's needed to understand what drives the tumor and what are its vulnerabilities. And based upon that understanding, it can either lead to rapid drug repurposing opportunities or new drug development opportunities. But ultimately, both of these paths converge on the need to test therapies in clinical trials. And it's important to recognize that because chordoma is an ultra-rare disease, there are some unique constraints that are imposed on running clinical trials. So obviously there are only so many patients out there, and that really limits the number of trials that can be run. And as a result, the bar really has to be high in order to justify the investment in a clinical study. Additionally, the disease is complex and heterogeneous, so not every patient will respond to a given therapy. And moreover, a patient's individual tumor is also complex and heterogeneous. So the response to a certain therapy is likely to be transient. So as a result of these problems of primary and acquired resistance, it's very likely that we're going to need multiple shots on goal here. And ultimately, we're going to have to run this path multiple times. And in order to do that effectively and at a pace that matches the sense of urgency felt by patients, we need seamless integration across the entire R&D continuum. But what we've found is that there can be multiple challenges to achieving this goal. For example, career growth and success often depend on the development of deep niche expertise in a specific biological pathway, technology, or therapeutic area, which, by the way, in many ways is really a good thing. But in the context of a rare disease research community, there just aren't enough researchers or realistic collaboration opportunities to ensure smooth mesh points along each functional area of this progression path. So academic labs can often navigate one or two of these gaps, but they're intentionally designed for functional specialization rather than running this entire gamut independently. Conversely, companies are structured for effective translation, but typically lack the pre-existing chordoma-specific experience and assays and research resources that are needed to justify an investment. So all this adds up to create several barriers to the effective evaluation and translation of new ideas for treating chordoma. I just mentioned on the previous slide how functional specialization can sometimes lead to inefficient translation. Researchers that are new to the chordoma field face significant startup time and cost barriers. There can sometimes be constraints on bandwidth or facilities that lead to concepts being picked up and then abandoned at various points. And many labs are interested in chordoma, but they're also interested in other cancers as well. And that can lead to lower experimental efficiency due to an inability to take advantage, ideally, of economies of scale. So our mission at the Foundation is to find a way to help bridge these gaps, to drive chordoma research forward at a pace that matches the sense of urgency felt by patients. And we've already managed to do this, I think, quite successfully for this last bridge here with our drug screening program, which was implemented in 2015 as a centralized and shared research platform that was accessible to the chordoma research community, leading to faster and easier and more economical in vivo drug testing. And this has turned out to be highly catalytic for us, resulting in 6 therapies being advanced into the clinic for clinical testing, or about one per year on average. But what we've recognized is that there are additional needs and opportunities for increased speed and efficiency across the R&D continuum. So, for example, related to resource development, there are needs and opportunities to efficiently validate newly developed models and then characterize them as well. Along those lines, I'm happy to share with you that we've recently completed a model sequencing project where we've profiled all of our PDX models and the vast majority of our cell lines using whole exome and whole transcriptome sequencing. That data is in the process of being posted in Cuvatica and in PDC Bioportal. So look out for a communication from me in the near future once that data is available. So there are needs for increased speed and efficiency in resource development, also in target discovery, where we can validate potential therapeutic targets and generate proof of concept data that's can induce or justify further investment. In drug discovery, we need to develop chordoma-specific assays that will allow us to perform experiments on emerging drug candidates, for example, targeting brachyury, where we can stand to more efficiently identify hits and then move to leads and progress those compounds forward faster. And then in translational research, there are clear needs for testing drugs in panels of chordoma cell lines that can serve as sort of a gating experiment that can justify further evaluation in our in vivo drug screening program, and can also inform the design of those studies. Additionally, translational experiments that can help flesh out therapeutic mechanisms and understand signatures of response and resistance can really help justify adequately therapeutic concepts so that they meet that high bar for clinical advancement that we referenced earlier. And so these needs can be addressed by a core set of chordoma-related research capabilities. And so to get inspiration for how to achieve this, we took a look at some of the most successful biotech companies. And what they have is core expertise in a specific therapeutic or technology, along with an ability and a laser focus to effectively translate that to the clinic. And then they search for the disease indications that can most significantly benefit from that therapeutic or technology. And we thought, well, maybe we could sort of flip that concept around and make the disease indication the central focus by establishing a centralized laboratory with proficiency in a core set of experiments related to chordoma, while maintaining a translational emphasis, right? And in that way, our hope is that we can complement the research community's specialized expertise in specific biological pathways in chordoma and in technologies and therapeutics. And really, the hope is that this shared research platform that we imagine can be a helpful resource that can get answers to key questions quickly and easily, so that we can help move projects forward, unlock additional grant funding, and expedite critical go, no-go decisions to ensure that chordoma research and chordoma researchers alike are successful. So certainly this idea is not without precedent. There are a handful of nonprofit disease-focused research labs that have proven to be highly catalytic in their respective disease areas. And so all of this led us to launch what we are now calling CF Labs earlier this year at the beginning of 2022. And we're located in downtown Durham, North Carolina, in a biotech incubator space called BioLabs. Some of you may have heard of BioLabs. There are a number of locations across the United States. And it's a shared lab space that provides access to state-of-the-art facilities with minimal upfront capital investment. So we've launched our 100%, our lab that's 100% dedicated to chordoma research at the beginning of the year. On the right here is a picture of me next to our scientist, Nindo Printuri, next to our lab bench. And at CF Labs, we have a flexible operating mode. We can provide basic experimental support, kind of analogous to a chordoma core facility. So we recognize that, for various reasons, good ideas are sometimes difficult or impractical to pursue. And so we'd like to try to help with that. So for example, maybe you're a data scientist or a bioinformatician that has just analyzed some chordoma sequencing data sets, and you found a new potential interesting therapeutic target. But you need somebody to help you validate that target. Well, we can take that on and we can help you validate that target in a set of cell-based assays. So we can provide some basic experimental support on one end of the spectrum. And then sort of on the other end of the spectrum, we do have the capability to engage in deep integrated collaborations that can sort of shuttle concepts through this entire pathway here. And we have the chops, I think, to be able to help you do this. We have experience in basic and translational research at top-tier academic medical centers, as well as drug development and platform development experience in industry. And we know how to move projects forward with the track record of contributing to multidisciplinary collaborative research teams that have published in top scientific journals. So we launched in January of this year, and the most notable thing that we did there was recruit Nindo Punturi to join as our first scientist. And this was really a huge win for us. Nindo has fantastic experience in lab startup and management, and really a rich scientific background in translational breast cancer research. And he's brought some of those lessons over into the chordoma field. And so Nindo was really our boots on the ground and allowed us to hit the ground running, so that by February, we were able to import 21 chordoma cell lines into the lab, stock and set up the lab, and begin establishing and optimizing our experimental capabilities, so that by the end of the first quarter of this year, we were really starting to collect our first data and initiate our first projects in earnest. So we have established and optimized a number of experimental capabilities so far, shown on the screen. And I just like to note that the protocols that we establish and optimize, and much of the results that we generate will be shared with the chordoma research community as soon as reasonably possible. And we'd really like to start a conversation about protocol optimization and best practices, and try to just standardize things to the extent possible to minimize lab-to-lab variability in results. So with all of that being said, I'd like to just take a few minutes here and provide a few vignettes for you of some of the projects that we have underway in the lab right now, and sort of apropos to what we were just discussing on the previous slide. The first example project I'm going to mention involves a protocol optimization related to our drug assays in the lab. So as many of you know, the accurate evaluation and comparison of drug activities requires careful optimization of experimental parameters that essentially dictate where you start the assay and when you end the assay. And that is particularly important for drug compounds that have a cytostatic mechanism of action. So this sort of optimization has never really been systematically evaluated and reported for chordoma cell lines, which creates the potential for lab-to-lab variability in assay results. So for example, one lab might find that drug X is sensitive in chordoma cell lines, whereas the next lab finds that that drug appears to be resistant, and that difference can be explained potentially by a technical issue. So we're optimizing parameters for each cell line. We're putting them in the IncuSite and imaging them as they grow, and we're sort of optimizing these parameters, and we're going to share them with the research community online as soon as possible in order to promote standardization. So in another example of a project that we have ongoing, we're really seeking to facilitate go, no-go decisions. So we've been working with a pharma partner to evaluate their asset, which we'll call drug A. And we had recently completed a series of studies in our in vivo drug screening program with drug A, and the results looked really promising and encouraging. And so we met with the key stakeholders and reviewed the data, and collectively there was a decision to compare drug A to its clinical stage competitors in preclinical models of chordoma. But establishing chordoma-specific experimental capabilities would require significant startup time and cost for our partner. So we said, hey, listen, we can do this in a matter of a couple of weeks. Let's set up an MTA. You can send us some compound. We'll run this study for you and give you the data back. So that's what we did. We compared drug A to 3 clinical stage competitors and 3 chordoma cell lines, and found that drug A compared favorably both in terms of potency and efficacy in these models, and that helped guide the next steps rapidly and efficiently. So in another example project, we validated a new therapeutic concept to essentially provide proof of concept data to hopefully justify further exploration in chordoma. So we had been working with a really fantastic research team over the past several years, and they identified a potential vulnerability to an atypical form of cell death called ferroptosis. But due to strained bandwidth and other competing priorities, they decided not to pursue this hypothesis further. We kind of told them that we were interested in evaluating it further, so they sort of gave us their blessing to adopt the therapeutic concept for further validation studies. So we sort of imported it at CF Labs with a view to enabling go, no-go decisions for further R&D in chordoma. And what we did was we targeted this pathway using commercially available tool compounds, and found that in a variety of chordoma cell lines, they were susceptible to ferroptosis at pretty relevant concentrations. And so we saw this both in our drug assays and in imaging experiments. And importantly, we also found that this effect can be rescued cleanly, supporting an on-target mechanism. And so the hope is that this data may support or induce further evaluation with an emerging class of therapeutics. And finally, the last snapshot that I'd like to share with you today is a project that doesn't involve a new concept, but it's sort of an internally driven translational research project that focuses on EGFR, which has long been considered an attractive therapeutic target in chordoma. And inhibitors of wild-type EGFR, such as afatinib and cetuximab, are sort of considered by us to be the gold standards against which therapies are compared to in our drug screening program. And both agents are currently in phase 2 chordoma clinical trials. But for chordoma, we still lack a good understanding of what predictive biomarkers are of EGFR inhibitor sensitivity, and also what the mechanisms of primary and acquired resistance are. And the idea is that this information can help guide personalized care by sparing likely non-responders the side effects and loss of time of an ineffective treatment, while also informing combination or sequential therapy approaches to address the problem of resistance. So we've begun initial experiments, basically testing all of our chordoma cell lines against EGFR inhibitors. And already what the early data are starting to show us is that we can resolve quite clearly sensitive cell lines and resistant cell lines. And what's really remarkable to me is that the most sensitive cell lines have EC50s that are comparable to what's seen in EGFR mutant non-small cell lung cancer cell lines, which represent the paradigm for EGFR inhibition in cancer. And so we're planning to grow this data set using our optimized drug assay parameters to 15 to 20 chordoma cell lines. And these emerging results are already prompting exciting and important questions, such as what are the mechanisms of acquired resistance to EGFR inhibition in sensitive cells? And what we've seen, for example, in UCH1 cells that have undergone acute treatment with afatinib, is that some of the cells quite clearly die, whereas others become enriched over time. These drug-tolerant cells undergo sort of a phenotypic transition to a more spindly mesenchymal type of morphology. And interestingly, these sort of lineage transitions are known resistance mechanisms to EGFR inhibitors in lung cancer and might point to potential combination therapy strategies so that we can reduce the sort of residual disease that might grease the skids for subsequent acquired resistance. Another question that is becoming interesting is what dictates whether EGFR inhibitors are cytostatic versus cytotoxic in chordoma. So we've already started to see a difference from cell line to cell line. So might it be the case that these sort of lineage transitions to maybe a more mesenchymal phenotype reduce the ability of afatinib to upregulate BIM or another BH3 domain family protein, and they suppress apoptosis, as is seen in EGFR mutant lung cancer, for example. Or perhaps they result in rapid feedback reactivation of receptor tyrosine kinase signaling that upregulates pro-survival proteins. So what dictates whether EGFR inhibition is cytostatic versus cytotoxic? If we can understand that, it might inform combination therapy approaches so that we can tip the scales towards cytotoxicity and deepen the magnitude of those responses. And then finally, why are some chordoma cell lines sensitive, whereas others are resistant? This question is very interesting to us, and we are working to create a predictive model of EGFR inhibitor sensitivity in the lab, taking advantage of our model sequencing data sets where we have genome sequencing and transcriptome sequencing, both on our cell lines and our PDX models, as I mentioned, as well as our emerging data set of drug sensitivity profiling with afatinib and cetuximab. And what we'd like to do is take this collective data and create a predictive model that can then be validated and refined in our drug screening program with our PDX models, and then ultimately and ideally validated in the clinic with patient samples. And so as far as this preclinical validation goes, we already have some really interesting data where in models that have matched PDXs, we've seen that the in vitro results correlate with the in vivo results. So in a cell line that is sensitive to afatinib, we see that sensitivity carry over in vivo. And in a cell line that is resistant to afatinib, in the cognate PDX model, we also see resistance. And so we are really interested in pursuing this further, but one thing that we do not have the capability of doing is creating this predictive model. So I would just like to highlight one potential collaboration opportunity. If anyone is interested in working with us on this to generate a model and sort of understand these the answers to these questions for EGFR inhibitor sensitivity in chordoma, I would love to hear from you at dan@chordoma.org. So a couple of other examples of ongoing projects that we have, just real quickly, we have sort of, we're evaluating a biomarker-guided therapeutic hypothesis in collaboration with a sarcoma medical oncologist who doesn't have a lab. And so this is kind of actually more of an example of a deep collaboration where we're also working with one of his academic colleagues who has expertise in this specific biological pathway, as well as the relevant experiments for probing that biology. And we are contributing to chordoma-specific research resources and expertise. And together, we're trying to understand whether this biomarker does predict sensitivity to a certain class of therapeutics in chordoma. So far, the results are looking incredibly exciting, and I'm looking forward to seeing where this project leads. We also have other projects ongoing that are better examples of CF Labs acting as more of a chordoma core facility. So we're performing in vitro drug screening experiments on behalf of a pharma partner to evaluate whether further in vivo studies are warranted with their compound. Is there a signal in chordoma? Should we explore this further? And then we're also helping some grantees validate a potential therapeutic target that they've discovered. So very interesting work being done there. So please do reach out to me if you're interested in collaborating at dan@chordoma.org. And we're growing. So we're currently hiring for a new full-time position with an emphasis on brachyury drug discovery and on evaluating immunotherapy concepts in vitro. So if you know anybody who might be interested in applying, particularly recent master's or PhD level graduates, please direct them to our LinkedIn webpage or send them to me directly, and I can point them to the application. And finally, I wanted to just say a word about confidentiality. From our perspective, obviously, sharing data and new discoveries is essential to progress. But we also appreciate and recognize the need for confidentiality in order to support the growth of your career and your business. And so, I wanted to mention that results from internally driven experiments will be made publicly available as soon as possible. As soon as we are confident in the result, it's reproducible, it's robust, we're going to share that data. But where we're collaborating with academic or industry partners, that data we will consider confidential until publication or public release of the data. But in any case, we think that these things go hand in hand. And our hope is that by facilitating progress, we can reduce the timelines to data sharing. We can expedite publication timelines, the approval of your grant applications, time to market, and ultimately, and most importantly, the impact on chordoma patients. So thank you so much for your time. We'd really love your feedback. Again, we'd like to, we've kind of, described our vision for the lab, but that's by no means set in stone. And we want to make sure that we're positioning ourselves to be the most useful resource possible for the chordoma research community. So we'd like to understand, where your biggest pain points are in preclinical chordoma research, what additional capabilities might be most helpful to you, and then, what's important to you in partnering with us? What do you see as the biggest risks or challenges? And then if there are any opportunities where we might be able to help you take advantage of research questions, please do reach out to us with those as well. So thank you everyone for your attention.
Maybe just a final comment, which is to say that, or just to underscore what the point that Dan made at the end, which is that this is really something that is being at the early stages of being built, that is continuing to evolve, and really is, our hope is that it is something that turns into a great value to many in the chordoma research community. So if there are needs that you have, if there are ideas that you have, if there are hopes that you have that the lab can help address, please don't for a second hesitate to get in touch with us. This is really, in a way, the chordoma research community's shared lab and a shared resource. So please help us make it valuable to you, help us make it what you hope it will be. And I think more than anything, we're just very appreciative of your interest and your continued dedication to chordoma research. If we can be helpful in any way at all, please don't hesitate to get in touch.